Well, good morning, everybody. I hope you've all been enjoying COFES as much as I've enjoyed being here. It's kind of a neat experience for a first-timer. We're going to be talking about patterns of life. And to kind of frame this a little bit, I'm kind of curious, how many of you, when you were growing up, grew up with this kind of vision? You wanted to go into space and to see the universe and to explore. That you grew up either reading science fiction or watching TV programs and, thinking of yourself as Spock or somebody else traveling around in the universe. I very much did, and, seeing Scotty was kind of a big start off for me, thinking, wow, if I become an engineer, I can go into space. It was later I discovered that was not for this century at least, but hopefully for future ones. I give a little bit of a tribute to that in the second part of it, The Life, the Universe, and Everything. You'll recognize that as the title of the third book in the Hitchhikers series. It was interesting. I'm a Doctor Who fan, and that was originally drafted up as a Doctor Who script. So I've always liked this idea of exploration and how science and the meaning of things went. And when I went to school and got my PhD, I started in working in numerics. And I, like most engineers who are in academia, kind of ended up in proving math and staying inside my office. And by a strange set of coincidences, somehow ended up becoming an explorer and making it to a bunch of these type of areas that I never would have expected. And it had to do with this idea of dealing with limited resources. And I know there's a number of people here that deal with that idea of sustainability, and that's a pretty big underlying concept in what we're going to be talking about. There's going to be some takeaways that I want to give to each one of you about kind of what I've discovered along this little journey, and hopefully my experiences will spark off your thoughts about where you might be able to go. So we're going to talk about saying yes to challenges and the importance of that, and ahas and the importance of networking, which you've got a great network going on here in COFES, but we want to make sure we expand this out to include lots of other things. The importance of usefulness actually to the creative process. How do we grow our technical? We'll talk a little bit about robustness and how we looked at adding robustness, how adaptability was added into our areas and what that means. And then how do we find meaning out of all these things? And I think that's really important when you're thinking about designing software tools. And a lot of the stuff we're doing is also developing software tools to help explorers and NASA be able to determine what is life when you don't necessarily know what that means. And this kind of all comes out from an area that, imagine yourself as NASA. We talked about, all the sci-fi and great things of that sort. Well, if you were NASA and you were going out looking for things, if you bump into Vulcans and whatever else, you're not going to have any trouble telling that that's life. If you go to a planet that's dominated by life, you're not going to have any difficulty recognizing it if it's just everywhere. The problems in figuring out what's life and where does it come from happens when life is scarce. And when does that go? And that's really what kind of started this whole process for us. It started in a conference, and it turned out that the lady sitting next to me was running an astrobiology institute in, of all places, Hawaii. And she was lamenting that she didn't have enough people who were going to it. She needed grad students. I was kind of shocked that she was having trouble recruiting grad students to spend the summer in Hawaii. But I turned to one of my grad students who was sitting right next to me. Her name's Jane Kernut, and asked her, hey, you want to go to Hawaii for the summer? And she was like, yeah. And so, and so I literally wrote her recommendation on a napkin, and she got admitted into the program. But part of that process was that for her to do that, yes, she had to answer some tough problems. And so there was that little bit of moment, am I going to be able to do this? Because that's not what I studied. And I told her to say yes. I love quotes. And Branson has a great quote on here that, if somebody offers you an amazing opportunity like this one was, right? And you're looking at this, you don't know how you do it. That is, say yes, figure it out later. I tell my students when they're in an interview, say yes, yeah, of course I can do that, right? You'll figure it out. If you've learned, humans only have a few ideas, right? We mix and mash them and call them different names and things of this sort, but we really have a few ideas. So if you understand those underlying ideas, the unreasonable effectiveness of mathematics is gonna help you out through this process. So you just need to figure out what it is. And there's another side thing that's kind of important to this. Since each of us comes from a different perspective, and our discipline brings a different perspective about what's going on, when you go and get involved in a project that you're not trained for, you bring a different way of viewing and understanding the problem, and that insight can really positively affect the situation. You don't want 20 copies doing the same task, right? If two people think exactly the same, one's unnecessary. Now, since I am a professor, I'm a full professor at Baylor, I feel an obligation at an early morning talk to give you some mathematics. Now, this was the challenge that was given to my grad students, right? It turns out that, grasses, this one is grasses growing in the Negev desert in Israel. And one of the Israeli physics professors saw grasses, and you could see it kind of burns in some strange patterns that's out there. And this happens over and over again. Patterns change year to year, but the basic idea seems to keep coming back. So he was curious, why does this happen? Being a physicist, he walked in and he said, okay, let me figure out what these are. We'll take population and the nutrients in the soil and available water, and we'll write a nonlinear partial differential equation in multiple dimensions and tune the parameters and see if we can actually account for this under these. Now, I'm sure all of you are looking at this and go, obvious. But believe it or not, when you hand this to a biologist to understand it, most of them look at you like you are crazy, right? And we can tell them things like, well, this is a growth term, it depends on water and nutrients that's available. And, this one allows for that oscillation and bias. You can kind of think of the grass growing here as like wave peaks. Right? So we need to account for that. And we've got a term that allows diffusion. The plants have to be able to spread across. And the nutrients, there's constantly nutrients being added from the soil, but the plants are consuming it. Right? And so you're running low. And the water, there's some precipitation. And then as anybody who deals with sustainability knows, there's a million ways that you can lose water from a system, from runoff and evaporation, absorption, and so on. Right? But even with this, to tune the equations to make it work takes a lot of training to be able to get what you want. And it's kind of more of a top-down than a bottom-up approach. You have to understand the whole system before you're able to actually do anything with it. And if you understood the whole system, it's kind of questionable why you're simulating it if you knew everything. And so the question is, how do you look at a system you don't know and pull it out? And so this problem was given to her by Chris McKay, who's a senior scientist at NASA Ames, and he wanted to know, could we find an easier way? And he wanted to, in particular, look at cellular automata. Now, cellular automata, right? What you do is it's kind of more like a picture, right? You have individual pixels representing locations, and you can, for each of those little boxes, you can have variables. So we can have ones for the biomass, we can have ones, for available water and all kinds of other good stuff in there. And the next state of your current cell for all those is dependent upon your neighbors. And you can make it the particular pattern of the neighbors or the total quantity. You can weight them different ways to account for gradients. Like, for instance, we can take one side of this and we can make it negative, and we can make the other side and weight it positive, and you'll get an idea of a slope. Right? So we can build all this in, but we can build it behind the scenes. So they don't have to look at it. And the prospective goal is to be able to get something that kind of looks like a picture, and maybe we can actually just take from photographs. So they don't have to try to guess on there. They can just take a picture of what they want, and we can deduce the rules that's going on inside here. So that's kind of the goal of what's doing of our project. And so we went and tackled this area. And this is soil crust in the desert. This is the Mojave. And you can see here these kind of dark areas. This is the actual crust themselves. Soil crusts are combinations of a variety of different organisms kind of living in a community. It's got lichens and cyanobacteria and all kinds of fun stuff. And each one brings something. Some of them bring solar protection, other ones store water, some provide nutrients. They're really important at fixing nitrogen into the soil, which is crucial for other plants to colonize deserts. They also hold the sand down, so they're really important for desert health and protecting the systems. And if you notice in here, they grow. You can see linear patterns and you can see the curves. You can see right near the edges where they are. They're a little bit thicker. And so the question was, could we use these to model these? So, this is one example that goes in. You can see on the edges, we get that little bit of thicker growth that's going on. And inside we get linear patterns and curves, and we're pretty happy with ourselves. And so we presented it at a conference and we figured we were done, right? Project solved, we had software, we knew how to do it, right? Clearly, what else could go on, right? And the project honestly would have stopped right there if it hadn't been for the networking and that kind of aha moment. And this is kind of that second takeaway point, right? Isaac Asimov, again, another one of those really influential writers in my life, and that has this kind of neat quote that it's not the eureka, I found it type of thing, I get it quotes that really matters. It's the, that's funny. And it's not always you that makes that's funny comment. In our case, the that's funny comment actually came from someone in the back of the audience. And her name is Penny Boston. She's now the head of NASA's Astrobiology Institute. And she goes, I've seen your patterns. Right? And we're like, oh, well, we're thinking, okay, well, we were modeling off grasses and soil crusts and that. She's probably seen that. She's like, no, I've seen them in caves. Right? Now, on the left-hand side, this is Cueva de Villaluz, which is a sulfuric acid cave in Mexico. You can see the dark parts of the living material, and the gray is the natural walls. You can see this kind of brownish area. This is mud that kind of seeps in from above on the roof. And they form these fascinating patterns that were basically pretty similar to what we were doing. This is a very different cave. This is Thrush Cave in the southeast of Alaska. Get a lot of rain in that area. And again, you can see it. This is over moon milk, which is kind of a neat substance that forms as a byproduct of all this. But again, we get these patterns that are growing. But it wasn't just there. Lava tubes. This is in Hawaii. They never invite me to these ones. I keep trying to figure out how to get invited to the Hawaii trips. But you can see along here, again, we've got the organisms growing all along, hanging off this smooth lava tube walls. This is in the Galapagos off Ecuador, right? And again, you see these organisms that are growing. This is kind of interesting because this is, again, Cueva de Villaluz, the patterns, but this is the twilight zone, right? Not like do. But the twilight zone is where there's enough light that photosynthesizers grow, right? So we're getting a different type of mechanisms. The ones that are in the deep cave are drawing their nutrients from the walls of the cave. These ones are able to produce their own. This is Salt Creek in Death Valley. It's a hypersaline creek. And this is on the bottom of the creek. This is the algae growing, again, in those same type of patterns, because it's dealing with this area where it's got highly constrained resources. Right? In this case, even though it's in water, the salt greatly affects its ability to grow and to maintain its health. So we took all this kind of into account and refined our models, and we were like, oh, great, we can model these by handling them, and we were able to meet a lot of requirements, and right, it was neat. It solved her problem, right? But this is where we started thinking, okay, if we can do this in all these environments, this is where that kind of usefulness constraint comes in. Because again, it was at that cusp. Is it gonna become just a neat tool that you can send it out and biologists can look at things, or can we go beyond that? A lot of times people treat creativity and usefulness as enemies. Dr. Amabile is a professor at Harvard, and she actually teaches in their business school. And she had this neat quote that really kind of resonated with me, which was, to be creative is linked with useful and actionable items. right? That's actually important. It's got to actually impact it, or it's not really truly creative. That there's not a separation between these ideas. Right? And for us, the creative side comes into, and the idea of usefulness kicks into, how can we adapt this tool that we had been working on to meet the needs of NASA? And I've already mentioned, when you're looking for life, it's the extreme environments that are going to be the interesting one. Think about Mars, right? I mean, if Mars had life, which I'm hoping it did, but if it did, it went through a period where it kind of died off. So when it died off or died down, if there's some still stuck in a cave somewhere underneath the surface, it went through a period of time when it was low on resources. So how do we actually take what exactly we're doing, which affects that area, to help them look for life and to make it useful to them? Well, here's where we kind of tied into things. We said, well, look, ours is potentially a universal biosignature. Why do I say that? Well, because the nature of the chemistry didn't matter. Most tests for life, I test for the presence of this thing. Do you have ATP? You know, is there this particular chemical process going on? Things of that sort, which matches great what we've got here on Earth, but how do you know that's what matches other things? There's a lot of chemical processes that could provide the basis for metabolism. We know of a few, and they're pretty tightly related, but it didn't seem to matter here, the chemistry, right? Sulfur-rich environment, carbonate environment, iron, manganese, heavy metal, right? It didn't matter. It didn't matter what the bedrock that it was on, right? So the geologists were always thinking, hey, maybe it'll vary by bedrock, but basalt, limestone, granite, different types of lava, didn't matter, right? It also didn't matter which ones were in there. So, you know, I mean, we had heterotrophs and prokaryotes, and you can just go down the line. You know, every branch of life was represented and did these things. You see it in higher plants. Human beings even cluster up. You look at how we tend to cluster and build things, and, you know, where we live, and, you know, you're not going to find a successful community that doesn't have parks, empty areas that are in there. These same type of patterns form within human society. So the nature of the organism doesn't matter. The scale didn't matter. Trees do this, right? It's also preservable. So Cueva de Villaluz is where we went on our National Geographic expedition, so I have a lot of examples from there. This is kind of living, growing bioverms, right? Bioverm life, and verm is worm-like, so it makes these worm-like patterns. So you can see them living here. This is a lithification front, right? It's the plants are dying, or the organisms, it's, you know, a wide variety of stuff from nematodes on up, are dying off. And what's happening though is their patterns are turning to rock, and they're going to be preserved for geologic timescales, which means millions upon millions of years later, you can still find these patterns. And these patterns have been found preserved 100-million-year-old rocks. So you could also see here, this is in a desert in Australia, you could see things growing on the bottom of the rocks, right? And the crystals provide kind of like a greenhouse to them, where it shelters them but provides protection for them. The question about sample return or sending it, well, if you send an astronaut out there to look at them, they have weight limitations on how much they can test. So even if you've got a human being looking for it, there's limits. If you want to do a sample and return, which would be the scientist's next favorite thing, you have to do something like this, but you've still got to limit how many samples are you going to pass back. You cannot sample everything. If you want to just let the test be done in situ, right, to send out, you know, your Mars Science Lab and let it test things, you only got so many chemical tests before you run out. So you've got a limited number of shots. You better make sure the shots you take are good ones. Right? We have the ability with a camera to tell you what's more likely or less likely. So, right, added on, what if you want to do autonomy? Right? Now you have to let probes kind of decide where the good spot is. The further out you get, the less likely it is to send back to us and say, what do you think? Right? So ideally, you know, when it's looking and trying to assess, should I go here? Should I go to this location? Should I go to this location? Which one's going to be more likely for there to be life? You want your best opportunity. Well, with the rule sets that we can develop by extracting them out, right, we can do stuff. We can figure out, hey, this is what it looks like. And we can categorize what the models for life look like. We can tabulate what those ranges are, which will allow us to then set a confidence interval on this. And then higher confidence would indicate good spot to go. We can adapt our tool to become useful to the project that needs to be solved. And that's a crucial part of this creative process. Now, whenever you're working with NASA, there's always this kind of question, right? Failure is not an option. I'd always heard that was what was said by the people in mission control, but it wasn't. This was actually said by the script writers. They went and talked with the people, and they kept hearing about the importance of never failing, and they came up with this great tagline, which they then attributed to them, and it's gone into the books. But this is really the idea of robustness. Right? We want to make sure, no matter what hits it, that we can handle a variety of stuff. Well, how do we find that? Well, we've got to take a look at what's out there. We've got to see where all the perturbations go. As engineers, we've got to characterize it. And by characterizing those things, we can then start to make models that go on. So for us, that meant we had to go into the field. Thankfully, NASA obliged. For those of you who like the education application of this, this is a really neat project. NASA's Spaceward Bound. They take a bunch of scientists like us and they send us out with school teachers and ones who are training, and they're our assistants. And so they get to learn alongside how to do actual science that they can then take back to the classroom and teach kids. So it's kind of a neat way of bringing back and doing them. So we were able to get a bunch of, wrong way. We were able to get a bunch of highly scaled up pictures, taking a look at these. We were able to see that, hey, there's washes and how's that affecting? You can see the growth regions. We were able to make predictions about, for ours, for the growth on the outside edge, it meant that there had to be a higher metabolism. So we predicted there was going to be more metabolism on the edge of the patterns. The biologists thought we were crazy about this, but here they are testing it, and they found out we were right. So it was very, very happy to us that our mathematical models that didn't include metabolism in there actually was able to predict metabolism. So it was a good confirmation. And a shout out to Ernesto Gomez, one of my colleagues who's worked on this with me for years and years. From all this, we started noticing that these patterns and shapes, even though they changed, there were some basic things that stayed the same. How do you keep them from staying the same? Or how to keep them staying the same? Well, we said, okay, look, if we've got, this is the region of our cellular automata, and this is the center square. If we consider this the feature that's inside, right? It can be completely contained no matter where it is, whether it's on the edge of the feature or in the center, right? It could be such that it's completely contained if it's in the center, but not when it's on the edge. And we went through a whole bunch of different examples. And we set the rules about where things had to grow and where they didn't, following a couple things. If it's not shrinking, at the lower edge, it can't be dying off, right? So the edge stuff, the lower bounds, no death, right? If it's not going to increase, we got to make sure at the upper edge of the density, it's not adding more stuff in. So this is just kind of some basic intuition on the human side, but that allowed us to get some really nice formulas off them. And we didn't want them to grow together, so past the upper limit, we didn't want there to be extra growth that's on. We then took this and went on the National Geographic expedition. And this is in Cueva de Villaluz, and this gives you an idea of what some of these look like. These are the pools underneath. You can see some gravitational streaking that's going on here because this stuff's really gooey and it's quite thick, which is kind of hard to imagine, but it's a pretty amazing stuff that's in there. I have to kind of mention this because my wife says I'm crazy for doing this. I'm allergic to sulfur, and this is a sulfuric acid cave, and you have to go in with these breathers, but if they ever get wet, the filters stop working. And so I'm thinking, I could die here. But the opportunity was too cool. I'd rather die doing something awesome than live not. So I think I survived. But you can see there's a lot of neat patterns. And while there's gravitational streaking, there's also some ones that don't do gravitational streaking. All right? And as we started looking and studying the images, this is from a roof area where they come in, we were able to refine our models a little bit more to say, hey, look, in order to cover this, we don't just want where the rules are. It seems that things are being pushed to certain densities. We kind of likened this to like gravity. So in physics, you have this idea of potential wells, right? So gravity is kind of a potential well that keeps in. There's forces that push you and keep you contained in. Well, if we consider this as kind of density axis that's going on, this is the low density side and the high density side. There's growth, right, which push us up, increase the density of life that's in that area. And we've got die-off rules that kind of take things that are a little bit too much life and kind of push them back. You can think in terms of sustainability. You get too many people in an area, it's not going to be able to support you. There's going to be forces that naturally restrict. If you got more resources available, you'll grow. But it's the width of those walls will determine how strong. So how thick are those? And are they straight, or do you have little plateaus in there? And by taking those set of rules in there, we were able to get kind of nicer looking models that could account for some of the other stuff we were seeing. We were able to add in nutrient growth areas because we discovered, hey, all the nutrients seem to either come from the rock, but there's also sources that leak in from the outside. So we added in some kind of leak-in sources, and we're able to get these patterns that changed but still obeyed the rules. So wide variety of stuff worked for it. So by looking at what was really encountered, we were able to make mathematical equations that could really kind of cover the breadth of what we were dealing with. This is when you have to hit adaptability. Since I'm mentioning quotes that don't necessarily always get attributed correctly, I'd always heard that, it's not the strongest that survive or the smartest, but the one that's most adaptable is attributed to Darwin. But it's not actually Darwin who said it. So it actually turns out to be another business professor, this one from LSU, who was summarizing Origin of Species for some people, and he said, this is the takeaway from the book. And I kind of thought that was neat that it wasn't him, but somebody else who kind of gave what I think is probably one of the most memorable taglines or takeaways. We looked at a bunch of different things to try to do this. I mean, we tried crazy amounts of stuff, right? Everything you could imagine, from swarm intelligence to everything. And we were always able to pull some things out. It was tantalizing enough. We knew we were close. But how do we actually get something that really can kind of cover it? And it was when we started looking at histograms, which are very useful in a lot of image processing areas and are just starting to get used now in deep learning networks as one of the layers, as a histogram layer that kind of captures bigger scope. The idea of a histogram is we take that center cell, it's either has living material or not, and we count all the neighbors. So in this case, if you count up everything around there, there's 15 neighbors to that center square, so it just adds 1 to that area. And we do that for every single square. This is a small subset of one of the images, and we get this distribution. And it turns out this distribution is proportional to the probability distribution for the system that generated it. So kind of contained within them is the underlying rules that developed them. This was the picture I had mentioned just a little while ago. We take this and we apply that same set of histogram rules to it, and we get a curve that looks like this. Right? Now the green is if the center was alive, and the red is if the center was not alive. Did the center square have biology or not in it? Up along the top, since this was a simulation, I know the rules. Die off, growth, stay the same. Die off, growth, stay the same. Sorry, stay the same. Growth, die off, and then up here I've got to stay the same. So you can see this is kind of the wells, so this is a growth rule pushing you down. So you can see where the wells are located. And we know something kind of neat. If you look at this growth rule, it corresponds to this one area where you only have live centers. If you look at the area where this is die-off, right, there's just non-living centers there. So if I were to kind of summarize it, I could say, hey, look, so here's a non-living centers, and here's living centers, and imagine this is kind of the curves that's going in. It makes sense, right? I can have isolation die-off, and there's kind of a limit on the lower edge, and I can have more dead centers than live centers, but it'll transition, and where that transition occurs, you would think would relate to something about the underlying rules that made it. So it wasn't too surprising to carry it up. But the question is, was I just lucky? Right? So since we can generate tons of different examples, we take this same thing, and we needed to do it for lots of time periods to see as the pattern changed, did the distribution change? So we kind of summarized it up. We made there's only death in this location. There's more death than life. There's more life than death. There's only life. We kind of picked those 4 things. And so this is for that same set just running over time. This is your density, how many neighbors were there, right? This is time increasing, the iteration. So each one of these, if you took a row, is essentially one of those histograms. I'm just showing you the life only. I put the rules up here, and the separators, I kind of just gave you colored bands to let you see. Where you see the gray, that means I don't have any data. Okay? The black means it's not life only. The white means, yes, it's life only. And if you notice, where I have a growth rule, I've got life only the whole way down. Growth rule, life only the whole way down. Now this is over time. There's some slight variation, but it's not very huge. Right? Changing pattern doesn't seem to matter. This is the life dominated, more life than death. You'll notice it's always on the upper edge of the densities and on the areas where it's flat, stay the same. If I look at the area where there's more death than life, again, on the flat areas, the bottom of the wells, left-hand sides. Always, always, always. Right? How about the death onlys? Well, again, right where there are die-off zones. Right? It's just barely catching it here. That's what's preventing it from going higher. Putting it kind of all together, we kind of get these pictorial areas, and you could see it exactly maps out the regions. So it wouldn't have mattered when you snapped your picture. I can use this one picture to fairly reasonable probability extract your rule set. Now the only question is, that's assumed in this, is I'm claiming I know the radius that I'm supposed to use. I use the correct histogram size, and it does a great job. Does it work if we don't? Right? Because this would allow me to adapt my system. I could use this histogram as my adaptation cost function to find out how to pick the right one to do. Well, let's consider a real live example. We turn it into a binary image so we can do our histogram analysis. And what I did is I measured manually what the feature size was. Feature size is the width of things, so I can kind of measure some things as a human. And I get about 30 when I do it. And then I started off with too small of a radius. Right? And you'll notice as I grow it, right? So this is at about a quarter, this is about a half the correct size. And the shape of the histogram is the same. But the second I get above the actual feature size, it changes. I did this one slightly above, but it actually doesn't matter. Once you get above the feature size, you actually get the correct feature. And even going to 2.5 times, you'll notice some key things happen, right? Here's a peak here, peak stays. And you can look all along this, these peaks still occur. And if you find the centers of them, it's exactly in the same spots. Once you get above, the features seem to be fixed. Right? That means I've got a way to adapt my system. I now know how to change it. I can start small, grow it up. The second I see the pattern alter, I know I've got the right feature size. I analyze the whole thing. I've now got a way to adapt my system to that situation. Right? And the advantage of an adapted system is it's highly tuned for my problems. So now I found the way that I could do it. And it just, I mean, it's grunt work, honestly. But, you know, this is where the fun part kind of comes in. So at this point, we hit something that I think Pablo Picasso kind of brings out very nice here. Talking about computers, he says they're useless, right? They can only give you answers. At this point, I can make a computer that can give you an answer. What's the probability? What's all the rest of this? This is where, you know, your tools give people the stuff, but how do you draw the meaning out? Okay? Well, let's take a look and see how well we can, as human beings, start to draw some meaning out by applying this. Right? We can take this. This is chemical morphogenesis. This was done by Alan Turing, which I hope most people are familiar with. You know, and he had a way. He was trying to figure out how biopatterns himself work, and he wrote a simplified set of ones that were based purely off chemical only. And he formed some patterns that are pretty intricate that's in here. And if we analyze it, we can see the same thing: death low, life high, boom, 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 until we hit the feature size above. Right? And you'll notice they switch their orientation, so we know that we've hit there. But notice something interesting. It's really narrow. And as you grow, it's really smooth. There's not a lot of spread on it. Life tends to give you much more diverse models. It's not so tuned, it's not so repetitive, because life needs a diversity of things to be able to adapt. You've got to have members of your society in different states to be able to adjust to what's going on. And I think that's kind of an important thing. It reminds you about the biological one that's going on. Again, look at the spread of it, and as I go higher, I get lots of interesting features happening, although the key things are remaining. It's more what we tend to see in life that's on here. Now, a computer can note those things, but it can't really give you the interpretations to it. Grace Hopper, she's the one, if you're not familiar with her, who invented the term bug in your code, okay? She is a brilliant person who is like a quote machine. So if you ever want somebody who said something profound, look her up. She's got tons of stuff. But, you know, she had made this statement that it takes trained people to really understand it, right? You're building tools to help people make decisions, right? That's augmenting them. That's the same thing that we're trying to do. How do we give this? But it's the people who are going to do it, and we've got to keep it in mind. So let's see how well this helps you. You're now a little bit trained. So I'm going to ask you the question. I don't give you the picture here, right? But I am going to give you the histogram, so you see I'm fair. I've told you the feature size. Right? And you grow, and again, you see the life, death, and you get right about where it hits. It becomes kind of the interesting pattern. The peak's under, and it doesn't change. What do you think? Biological, abiological? Right? Yeah, it seems to have a lot of breadth to it. The pattern seems to get, you know, those kind of characteristics that happen. Well, you just analyzed that lava tube picture we saw earlier, right? It carried exactly that same type of one. Well, you're a little bit too good at this, so I'm going to give you a harder problem. Okay? So here's another one. You can see it kind of grows, but now, right, it definitely seems to carry forward. I'm not telling you the feature size on this, but right, it's narrower than what we're used to seeing. What do you think? Yeah, this is kind of that interesting stuff. It's mud cracks. Now, the neat thing about mud cracks is mud cracks form where there was a lot of water standing for a while. There was biology that was there. And that biology forms fibers that interconnect them. So the cracks are actually based on a combination. And even though the life's not there, so it's not forcing and keeping it robust, you can still see some of the remnants of that original kind of diversity that's carrying in and holding the mud together, right? Well, this is really kind of what we were hoping for, that you can even see the traces even when it's kind of questionable about what was going on, right? So this kind of made us happy, right? We as human beings can look at this. A computer couldn't have told you that, but as human beings, we can look and we can kind of understand where it's going and therefore make some conjectures about what could be happening here. So we're winding up here, so let me just remind you of our kind of takeaways from this strange journey I've been on, right? Somebody gives you a great opportunity, take it. You see an awesome opportunity, push into that area, right? Keep those networks up. Look for those aha moments. It's sometimes from very outside, right? Remember, creativity and usefulness are not enemies. They're actually really good friends, right? You got to figure out how to make your application robust, which means you got to understand the area so it can handle whatever's going on. But adaptable solutions, one that can tune themselves, are going to give you your best bang for your buck when you hit really challenging problems. And then in the end, it's the person who draws the meaning out. So how do you draw that meaning from the context? You need to provide tools that allow them to make those insights. So they're not doing the drudgery, they can think about the ideas behind it. I owe a lot of thanks to people. I've mentioned Penny Boston from NASA's Astrobiology Institute, formerly New Mexico Tech, Ernesto Gomez, a good friend and colleague from Cal State, Rakesh Mogul, who leads a bunch of the Spaceward Bound, and Chris McKay, who started Spaceward Bound, a bunch of other friends who have helped on these, as well as grad students, Jane Kernut, who kind of started this, and on down, Brian Strader, Melissa Quintana, Blake Schultz, Iwan Sanjaya, Zonki Richie Kai, Soli Deo Gloria. I'll leave you off with one final quote because I love quotes. This one from Ada Lovelace, right? I'm never satisfied. We should never be satisfied with the knowledge that we have. And so we should always be looking for knowing more. We only know a fraction of it, but that's the joy. There's great questions out there. Whenever you see problems, right? Problems mean it's a chance for us as human beings to really engage and to learn something new and grow. So if you have any questions, I'm happy to try to answer them.
So Keith, that was brilliant. Thank you very much. Just one very simple question. Have you tried applying this analysis to the astronomical structure of the universe?
We have not. We have thought about doing that, right? There's the kind of structure of dark matter, and if you read a lot of stuff in it, there's like the axis of evil and all kinds of things, right? There seems to be this kind of galactic, right? Axis of evil just kind of sounds cool. But it's really, there's some larger structure that goes into what's on. We have not used it to try to analyze it, but it would be an interesting one to take to see, is this kind of a chemical basis, or can we try to extract out maybe predictions about where patterns go? So hopefully if we get more funding on this, we've had some NASA funding, but always a challenge on that end.
Mr. Rain.
This is wonderful. Thank you.
My pleasure.
Have you had any discussion with Wynton Marsalis or other people in the Dixieland Jazz venue? Because they work these same patterns, but I don't think they know you.
There's actually several different groups that have kind of picked up. There's you could see some tackling of some of these type of problems going from the early 80s. Most of it has been pretty happy with where I said our project would have stopped if Penny Boston hadn't pushed me hard to kind of drive it to the next level. Where you kind of model it and you go, wow, it's neat, and I can see, I can do what I want. It would kind of be like your CAD tools or whichever tools you're designing, right? If people didn't drive you, I need this, it stays. And what I've seen very common out there is that, can we model it? Can we simulate it? Right? But I'd love to meet them.
Keith, I got a question on, and actually, I'll hold my comment. Okay. So in terms of where you're headed, we're seeing literally new forms of engineering and design. The thing that I'm curious about is, are those new forms of engineering design and the way, like when you're coming, like this is a different, there's a layer of tooling here that's interesting, different, and new, but there's a lot of things that are all the same. So, like we here in the room are designing tools that are already using many of the, those are good computer science principles, but you're applying it to a fundamental new area. Do you have comment on what that means, what the implications of this are for discovering new applications of the skill sets of the toolsmiths in the room?
It's an interesting, and it gets kind of to that very human level. And a lot of this came because I try to analyze my own self when I'm looking at it. And that's one of the things I'd encourage all of you to do is to turn the microscope back on yourself and see, what your tools and your thinking process says about you and your process that's going on. I think to each one of us, there's a few ideas underneath the sun, right? And a lot of what we're doing, 3 dimensions mathematically, you can show no real new phenomena happens at higher dimensions even if you mathematically model them. It's the same basic stuff. Differential equations, certain rules. So in some sense, we're kind of looking at different ways to tune these things to fit our users. And who is our user at the moment, and where are things actually being applied? And I think that's really a big part of this process, is how to get tools that work alongside human beings and use the machines where they're really good. They can calculate things really quick, but they're dumb, right? and we're really good at kind of drawing insights out, but we are not great at doing all those calculations. And I think finding that balance is really kind of the crucial aspect in those areas.
So it's a very nice presentation. So thank you for doing that. I'm a professor in statistics, so I'm going to ask you a statistical question. So it seems to me that mathematics you are doing, basically using idea of histogram, particularly also use idea of the neighborhood. This one very much related to something called nearest neighborhood in machine learning approach.
Yeah.
I'm just wondering, if you think about when you do the neighborhood kind of smoothing, the size of neighbor really matters, whether you have 3 neighbors or 5 neighbors. I'm just wondering for your analysis, do you look at sensitivity about how many neighbors you have in your analysis?
So that was kind of, I hinted at it briefly just to cover everything. I only had really one slide, which is when we grow the regions and take a look at different neighborhoods, and there's different types of neighborhoods you can do, right? There's more neighborhoods and so on and things like that. So we tried a whole bunch of them, but it turns out that once you get above a certain level, certain features become fixed. And those are the features that were crucial to us. Other things can slightly vary and alter, so we can actually adapt through starting at smaller neighborhoods and grow up and then watch when you actually get a fundamentally different distribution than the undersized. So undersized distributions will always form that kind of lump to the outside areas and split at the middle, right? Until we hit the correct size. And then it assumed the distribution that really was underlying it, at least on all the ones we've tried so far. But yeah, we do actually look at different sizes in that, because I think you have to. If you're going to adapt stuff, you have to have something that's you're sure is going to work. So, but we're constantly challenging it. Right? The whole principle of science is you should never believe that you're right. You believe that you haven't been disproved yet. So.
Thank you.
Let me, last question.
I had a quick question on, can you know what it is based on the histogram? Like, is the histogram a signature for the surface that you're analyzing? And if so, how could that be useful in analyzing surfaces that you don't know?
So the histograms are characteristic of a differential equation that described that probability distribution. So what we were trying to use them for is to extract out what the rules are, right? Now those rules could come from a lot of different things that will generate the basic partial differential equations. Our belief then is there's a second layer that's needed, which is that you now have to take a look at what are the different rules that are out there and which ones come more from biological versus abiological processes. And then we have to now do correlations off of those to be able to extract out where it is. But as a human being, when you look at them, you can kind of see, that's why I was giving you the couple tests. What do you see? You can see features, and those tend to indicate that at least there's something there, right? Which we then use on the correlations. And I'm careful not to put too much of that because that's where the next couple papers are coming from. So you can't scoop yourself.
Professor, are you going to be here the rest of the day?
I will be here till just before dinner. My plane leaves tonight.
Because
so.
there's a lot of people that want to talk with you. So thanks again. You're going to be here through lunch. You're going to be in the afternoon.
Yeah, I'll be here through the afternoon.
Thank you so much. It's so thought-provoking.
Thank you.